Welcome to another episode of the Your Longevity Blueprint podcast. Today, my guest is Kiran Krishnan, who is trained as a research microbiologist. He left academic research to help bridge the translational gap between research and commercial innovation. He's held leadership roles in international health and wellness companies. He has also founded and scaled research and technology development companies, including, most recently, Microbiome Labs, the preeminent microbiome therapeutics company in the U.S. integrated medicine market. Kiran has published numerous studies in peer-reviewed scientific journals and holds international patents in his name. Kiran is a frequent lecturer at nutritional and medical conferences, and he is a sought-after expert on human microbiome for health programs. Kiran is currently the general manager of Novozymes, One Health North America, and is a science advisor to 5 other companies in this market. Hopefully I pronounced those all right. Welcome, Kiran.
Thank you so much for having me. You pronounce all of it perfectly, so thank you.
Well, thank you. So in chapter 1 of my book, Your Longevity Blueprint, I'm comparing the foundation of the home to the gastrointestinal system in the body. And I'm sure you would agree that the gut is our foundation of health. And so today's show, I really want to focus on that, improving gut health for whole body health. Mm-hmm. So tell us your story kind of before we get into that. Specifically, why did you start Microbiome Labs?
Yeah, so I've always been a bit of a maniacal thinker, right? Just always thinking big in terms of anything I'm interested in. And so I've always been a big science nerd as well. I've had a deep curiosity to understand the world that surrounds me and the way things work. And so going into the science was a very natural reflex for me. My parents are both scientists in one way or the other. My dad is a microelectronics engineer. My mom's a medical doctor.
No pressure.
Yeah, exactly. No pressure. My dad, as it turned out, had over 200 US patents in his name.
Wow.
So almost anything electronic he's invented something in it. So he's got that kind of hard science, mathematical, innovative thinking. My mom has the soft science, the biological side, the clinical thinking, and I feel like I got a little bit of both. And I like thinking out of the box. I like systems biology, similar to an engineering view on things. But then I'm very clinically oriented because I'm very keen on impacting people, right?
Yeah.
And that's why I left academic research and all that to do what we do. We started Microbiome Labs purely because there was so much happening in the world of the microbiome, and there were things that were being discovered that could be so impactful to humanity. But what tends to happen in academic research is that most of the research just carries on for research's sake and goes deeper and deeper and deeper into a subject. Not a lot of it gets translated into things that actually impact people's lives.
Like clinical practice and how do I apply that as a provider? Yeah.
That's exactly right. So that's actually called a translational gap in science, because research and academic and research institutes are done for research's sake. And then there's some companies and some people that come along that bridge that gap by taking understandings and technologies and creating real world tools and products out of it. Sure. Microbiome Labs existed for that reason because of my megalomaniacal thinking. My view of being able to help large masses of people was about modulating their guts. And if you can modulate the gut in the right way, you can help tons and thousands upon thousands of people in a relatively short time. So that's where we started.
It kind of leads to my next question, because I kind of want you in your words to explain what the gut-brain immune connection is. Like, why does gut health matter when you're saying I can impact thousands, millions of people by improving their gut health? How? Why? Can you kind of share with our listeners, break that down more simply?
Yeah, absolutely. So there's a couple of different perspectives on this, right? And I'll give you the first and the most kind of one-dimensional perspective, which is correct, but it goes deeper than that. So number one, it's the gut being the digestive system, right? That's where we break down and assimilate and utilize nutrients that are coming into the body. There's very few nutrients that are produced outside of the gut. Vitamin D is a good example of that. That's through the skin, right? But there's very few essential nutrients and all that are produced outside of the gut. So at the most fundamental level, you need a healthy gut to be able to access nutrients from food and then feed the rest of the body these critical nutrients. That's the most one-dimensional way of looking at it. Here's a more the more three-dimensional and four-dimensional way of looking at it that actually is much more profound, right? Number one is the microbes in the gut constitute the largest component of our genetic capability, right? We have 150 times more microbial genes in our body than we have human genes. And our human genes are wholly inadequate to function, right? We have somewhere around 22,000 functional genes, which sounds like a lot if you don't know about genetics, but an earthworm has about 22 or 23,000 functional genes, right? So we're not that much more sophisticated than an earthworm when it comes to our own capabilities. And yet, in your gut, you've got over 2.5 million microbial genes in the system. So you have 150 plus times more microbial DNA in your system, and that DNA codes for proteins and enzymes and peptides and all these things that we need to function to be human, right? So our gut is the largest reservoir of capability of being.
I like that. Yeah, reservoir of capability. Yeah.
That's our and this is the treasure trove, right? This is what our ancestors went through hell for to preserve and build to pass on to us, that we are stewards of, and we're harboring this amazing ecosystem that over hundreds of thousands of years we've developed a symbiotic relationship with, right? And a mutualistic relationship with, that provides us functionality. We now know, you can look at, even at the lowest estimate, if you pull out a sample of your blood right now and you look at all the compounds that are in your blood that are important for cellular function and immune function and brain function and all that, more than 50% of those compounds are derived only from the microbiome, right? Meaning you can't get them from food, we can't make it ourselves, we can only make it in the gut microbiome. And if you don't have the organisms in the gut that make that compound, then you're in trouble, right? You lose that functionality.
Can you give an example of one of those?
Yeah, absolutely. So there's a number of examples, right? So the most basic one is something called short chain fatty acids. So butyrate, propionate, acetate, right? Those are the most well-known short chain fatty acids. Let's just take butyrate as one of them as an example. If you don't have enough microbes in your gut that make butyrate for you, then some of the things that start to get dismantled in your gut are your metabolic response to food, right? So for example, when you eat food, and one of the key things that's supposed to happen is the food starts getting metabolized in your gut, and some of the microbes metabolize components of the food, like the carbohydrate components, and produce butyrate. The butyrate binds receptors in the lining of your large bowel, and the binding of those receptors sends a signal to your brain that says, stop producing the hunger hormone, we've got enough food, start producing the satiety hormone called leptin, right? So now you're done with food, you can go, okay, I'm satiated, I'm finished eating. And at the same time, that signal binding of the butyrate to the receptors in the gut are also turning on metabolic pathways like cyclic AMP, which actually stimulate all the cells in your body to start burning fat for fuel, right? And they start telling your brain to start reading the blood sugar levels and controlling blood sugars by balancing insulin and glucagon and so on. So all of the metabolic processes that are supposed to happen in your body in response to food is dependent on the production of butyrate. And if you don't have butyrate being produced adequately, what happens is you get something called leptin resistance, which leads to obesity. You get insulin resistance, which leads to diabetes. And then both of those things lead to chronic low-grade inflammation, which leads to hypertension, heart disease, dementia, Alzheimer's, everything else, right? All from not having enough butyrate in the system. So that's just one example. Another great example I love is something called urolithins. Urolithins are these very unique compounds that actually help turn over dysfunctional mitochondria in your cells, right? So all the cells in your body have these little engines in them called mitochondria, except for your red blood cells. Of course, they don't have mitochondria, they have hemoglobin to bind oxygen. But outside of that, every other cell in your body has mitochondria. And depending on the cell, it can have anywhere from, a few hundred mitochondria to a couple million mitochondria. Right? If it's your heart cell, for example, or your glutes or your quads, these big muscles that have a lot of energy demand, or your brain cells, you're going to have a lot of mitochondria because those cells demand a lot of energy. The mitochondria is what produces ATP, which is the energy source, right? And the one of the most clear definitions of aging and this program being the longevity program, right? This becomes really critical to your audience. One of the most clear definitions of aging is something called the mitochondrial free radical theory of aging, which shows that age is defined at the cellular level by the number of dysfunctional mitochondria in your cells, right? If those engines in your cells start to break down, and they used to produce 27 ATP from one glucose molecule, and now they're producing 14, right? The functionality of that cell becomes compromised. The organ that the cell makes up becomes compromised. Eventually, both die off, right? And that's where you get degenerative conditions, which is what aging is, right? Degeneration in your skin looks like wrinkling and softening of the skin. Degeneration in your liver means you're not producing enough bile and you can't, digest food and absorb fat soluble. Degeneration in your heart means your cardiac output is going down. That means your heart can't pump enough blood to your system. All that is aging. Degeneration in the brain means your memory slows down, your capabilities slow down, right? All that is defined by the mitochondria being compromised, the cells dying, and thereby the organ system dying. Now, we have a system to regenerate the mitochondria, to fix the broken engines, and that requires this compound called urolithin A. The thing is, we can't make urolithin A. We can't get it from food. So where in the world does urolithin A come from? This critical compound that fixes the engines of our cells. It comes from certain microbes in your gut converting polyphenols into urolithin A. Right? We've outsourced that absolutely critical function for survival to microbes in the system, right? It's mind-boggling when you think about it. But if you think about evolutionary biology and the fact that we only have around 22,000 genes, you start to understand that, okay, we don't have the capacity to do half the things we need to do to be human. So we have to outsource those functionalities to our ecosystem, our microbes. And that was a critical part of the development of mammals, was the incorporation of a large number of fermentive microbes in the digestive tract. Right. And that became a key step in us moving up the evolutionary ladder and up the food chain, because that allowed us capabilities and resilience that most other animals in the animal kingdom did not have. Right. So that's why the gut is so important.
Yeah, long, awesome answer. But we need short chain fatty acids. We need butyrate. We need this urolithin A from polyphenols. It sounds like. Did I hear you right? That we convert. Okay. Let's simplify things for just a moment, or maybe we're not going to simplify things. Let's talk about probiotics.
Yeah.
So can you expose some myths? Most of the listeners have heard of pro and prebiotics, but can you expose some myths kind of surrounding traditional probiotics? And I know a highlight of your company is spore-based probiotics. So can you kind of differentiate the two and kind of explain when we should use spores, if there's any times we shouldn't use spore-based probiotics? Just kind of go down that.
Yeah, absolutely. I think one of the biggest misnomers about probiotics is that, it's just the more is better, right? More strains, more CFU counts. That's why I started seeing the industry going where they're going 50 billion, 100 billion, 200 billion. And in fact, there's zero science behind any of that. There are many cases where a single well-researched strain that does something specific is going to be far more powerful for your system than 15 strains that you don't really know what they do, let alone know what they do in context of one another, right? And so development of probiotics have occurred in kind of a vacuum of information, where the idea is that let's just throw as many things together as we can, and the volume is the key, right? When it's not. What we've come to know as we got more sophisticated in researching probiotics and understanding what these microbes do, you come to figure out that the functionality of each individual microbe is critically important when you're thinking about formulating a probiotic, right? It's not just about numbers. It's never actually been about numbers. It's always been about the specific capability of the microbe, right? Now, the numbers game can actually be a detriment. There's something we've been discovering over the last year and a half in the research we're doing with the APC, which is the biggest microbiome research institute in the world, that's based in Cork, Ireland. There's this concept that probiotic microbes are either quiet or they're loud. And what I mean by that is it's quiet or loud in context of your immune system, right? And what we're finding out is that the vast majority of conventional probiotic strains we're testing amplify inflammatory responses in your immune system, right? And it makes sense because you think about the vast majority of probiotics people have access to, none of those strains are native to your system, right? Which means that if you consume them, your body recognizes them as foreign, right? And because it recognizes them as foreign, your immune system is going to mount a response against them when they come in. And the response is going to be bigger the more of those strains there are, right? So the 100 billion CFU probiotic product is going to elicit a bigger immunological response than a 10 billion CFU probiotic. The thing is, the innate immune system is the first part that responds, and the innate immune system upregulates inflammation. So you can actually measure and see each of these strains when you introduce it to the system, inflammation goes up, right? Now, in a healthy individual, if you're perfectly healthy and all you're trying to do is stimulate your immune system a little bit, it's cold and flu season and you're like, I want to irritate my immune system so they're more active in case I encounter the cold or flu virus or whatever, that kind of approach may be fine because what you're doing is you're amplifying immune response, right? But you're amplifying an inflammatory response. So if your condition is that you are already inflamed, meaning you have a primary gut issue and immune issues, autoimmunity, you have obesity, you have diabetes, you have IBS or SIBO, any of those things, the last thing you want to do is amplify inflammatory responses, right? So getting into that kind of specifics, into the functionality of the strains becomes incredibly important for how you use probiotics. And that hasn't happened at all for the vast majority of the industry. And so I would distinguish probiotics more so, not as much between spores and non-spores, and I'll talk about why spores are so useful. But the bigger distinction is researched and non-researched probiotics, right? Because that is really the key. Like if you don't have studies on this 15 strain combo, you have no idea what that's doing in the body. And we know that many of these strains can cancel each other out or in fact be antagonistic to one another, right? We saw that in a study we did. We have a bifidolongum strain that's in our product called ZenBiome. This strain is incredibly powerful, has 8 published studies on it, right? And we show that it dramatically reduces stress. It dramatically reduces cortisol response when you encounter a stressor. It dramatically improves sleep.
I need this. I just got an email about this product.
Oh my God, it makes such a difference, right? It changes brainwave activity where it puts you more in a theta wave band, which is the meditative state when you encounter a stressful situation, right? It's a phenomenal strain and it's so powerful. What makes it so unique and why it's not loud to your immune system is because it's got this exopolysaccharide covering to it. The exopolysaccharide covering actually gives it a recognition by dendritic cells that'll actually engulf it in your gut. And then it'll metabolize part of the strain and spit out something called peptidoglycan. And it's these little peptidoglycans that modulate all the inflammatory response, the cortisol response, and all from stress and anxiety, right? We have depression studies, low mood studies, anxiety, stress-induced studies, and so on. And it's so powerful. We mixed this strain with one other bifidobacterium strain, a commonly used probiotic strain, and it knocked out the activity of the EPS strain, right? You would think, oh, we'll just mix more bifido and more.
Right.
They're all good for you, right? And it's better to have more. Nope. In fact, when we added other strains to it, it completely knocked out this strain's activity, right?
That's so interesting. Yeah.
People don't look at that stuff because companies aren't doing the research. So
can you say what strain this is again? I missed that. So this ZenBiome is the name of the product. And which which strain is this?
It's called Bifidolongum 1714.
Okay. So is that a psychobiotic then?
It's a psychobiotic, yeah.
Okay.
I like the gesture with that. It is a psychobiotic, yeah. And I love that term. In fact, the researcher and the clinician that came up with that term, his name is Ted Dinan. He's a psychiatrist, but probably one of the most innovative gut-brain researchers in the world, one of the most celebrated and published. And he works with us. He developed this strain with us, and he came up with that term psychobiotic. But it's a very, very powerful strain. And when you think about how it impacts our ability to modulate stress, you start to see that in certain cultures and certain types of groups of people that are living more native lifestyles than we do, they tend to have high levels of these types of organisms in their gut. right? Because they live a lifestyle that is actually more risky than we do, right? Because we have abundance of food. We don't have predators chasing us. We still have hunter-gatherer tribes in Papua New Guinea and in Tanzania and in the Amazon that live a life where they could be eaten by a predator, right? They have to go hunt and forage and gather food on a regular basis. And these types of groups of people have very high levels of these types of protective psychobiotic strains because it's key to be able to modulate stress response in these environments, right?
That's really cool. Yeah.
And we've largely lost it. This is why a single stressor, first thing in the morning, for example, can keep you at an elevated anxious state throughout the day, because you will go through this elevation of the fight or flight response from that external stressor, but it becomes really hard to come down from it because you don't have enough of these microbes in your gut. These microbes are the things that bring you down from the flight or fight response, right? So that's an example of a well-studied, well-understood and characterized probiotic. And with this probiotic bacteria, I would use it just by itself, right? Because there's no benefit to adding a bunch of other stuff to it, like other strains, to it, because it by itself is so powerful. And so that's where the research comes in. Now let's talk momentarily about spores. So why we went to bacterial spores is because what we were trying to do is mimic our interaction with microbes in the natural environment, right? If you got a probiotic product, and I looked at a lot of these and we studied, we tested a lot of these products, the common ones you see in, even sold through the practitioner marketplace. You take one of these, it's 50 billion CFUs, it's got 15 strains in it. My question was always, where in nature do we encounter this on a regular basis, right? Is this a natural type of exposure? And if it's not a natural type of exposure, then you have to understand and believe that your body's not going to react naturally to it. Right? The reaction and response to 50 billion of these strains coming in at once is going to be an unnatural response. And that unnatural response is likely not going to be favorable over time. And we saw that, and we see that now through the inflammatory responses that you get from certain probiotics. So we said, okay, what is our natural relationship with bacteria? So once we're born, right, in utero, we get some exposure to microbes. Once we're born, we get a huge inoculum through the vaginal canal. Hopefully, if you came out of the vagina. If you didn't, then of course you're already on the back foot, right, because you didn't get that large exposure during birth. And then breastfeeding, breast milk contains lots of different microbes, skin-to-skin contact early on with mom and dad. And then from that point on, it's the environment, right? The environment is where we encounter the vast majority of microbes. Now, most microbes in the environment aren't going to function as probiotics because they're going to die in your mouth, in your upper gastric system, or even in your small bowel, right? Bile acids and so on will kill them. But as it turns out, there are some microbes that exist in the environment. They don't live in the environment, they exist in the environment, that can actually survive through this antibacterial gauntlet that we have in our system to make it to the intestines alive, right? And so the ability to do that is a very unique ability for these microbes, right? This is an ability that nature has provided these microbes for a reason. So we started looking at these microbes and what they actually do. As it turns out, they are incredibly protective of the microbiome. One of the key things that the spores do, which is why I love them, they're so fascinating to me, is they go into the gut and then they do quorum sensing, which means that they read all the microbial signatures, right? And they can identify pathogenic or dysfunctional organisms. And when they identify these organisms, they'll actually sit next to them and bring down their levels. Then they can actually produce compounds to grow your good and beneficial organisms. So they can completely modulate your microbiome in a way that even we can't do it. That's another exercise, and a very important exercise that we have outsourced to microbes in the environment and our microbiome that we can't do ourselves. Right? So we're doing that. That's happening constantly in your gut if you gain exposure to these microbes. So that's just one functionality of spores that I really like. That's completely different from most other probiotic bacteria.
And is it true that spores can kind of change the terrain in the gut versus other organisms that last maybe 1 to 2 weeks and then are gone? Is that another strength in
They do, absolutely. And they change the terrain in a few different ways. Number one, because they can modulate immune response against other microbes, right? For example, they can identify a pathogenic or problematic organism in your gut microbiome. They can sit next to that organism and actually flag your immune system to recognize and deal with this organism, right? Then your immune system can produce antibodies like secretory IgA, for example, or antimicrobials against that organism to control the growth of that problematic organism in your terrain, right? They can also produce compounds themselves that target certain organisms, and they modulate certain aspects of your immune response, especially something called toll-like receptors. Toll-like receptors are these receptors on your intestinal epithelium cells and your immune cells that help these cells identify problematic organisms, right? So they're increasing the monitoring, they're increasing the scope and the understanding of the microbial environment for your own intestinal lining cells and your immune cells, and they're directly impacting the presence of pathogens. I'll give you one cool example of that. We did a study with Cleveland Clinic on C. diff infection, right? And we know that C. diff is a very difficult pathogen once it's established itself. People go through multiple rounds of antibiotics and have bloody diarrhea and all kinds of messy things. We started looking at whether or not our spores and Mega Spore can actually identify the presence of C. diff bacteria and what they do about it, right? And so, sure enough, in the study, we saw that the spores in Mega Spore can identify C. diff in the gut. So you have these areas of infection of C. diff. Our spores can actually figure out where the C. diff is and go to it. Now, Clostridium is a spore bacteria as well. So normally our bacillus spores will make antimicrobials to bring down the growth of these problematic organisms. But the Clostridium is probably more resistant to that than other organisms because its ability to form spores, right? And so the bacillus have found a new mechanism to go after them. And this is the fascinating part about it, right? They surround the Clostridium and then they produce chelating agents to steal minerals away from the Clostridia, especially iron. Clostridia and many pathogens require iron in order to stimulate their metabolic functionality and produce toxins, which is what they do, right? This is why bloody diarrhea exists with Clostridium infection, is because they're eating past the mucosa to try to get to the blood supply, right? And they're causing bleeding in the gut for that reason. So these spores are so intelligent that they sit around it and they chelate the iron away from the Clostridia and starve it to death, right? It's fascinating, mind-blowing.
It is, yeah.
Right? This is nature at its finest. If you gave me all the money in the world and the smartest scientists, in 100 years, we couldn't design a microbe that does that, right? This is an illustration of how amazing and complex nature is. We just have to be smart enough to know it's happening and take advantage of it, right?
So,
we can't engineer this.
So since we're not engineering this, where are these coming from? And just for the listeners, I just want to point out, so again, Mega Spore Biotic is is your main probiotic that is spore-based. So when you're referring to the bacillus strains, so those are the strains in specific product from Microbiome Labs. Just trying to clarify this because it can be kind of confusing. But where do the spores come from? Like, what's the source? What?
So they were originally isolated from healthy human volunteers. So we were looking for, actually, we didn't do this work. This was done by Royal Holloway, University of London, preeminent researchers in the spore world. They were looking for human-derived bacillus endospores, right? Because you can find bacillus endospores everywhere. They're ubiquitous in the environment. You find ones that are specialized to live in the ocean or specialized to live in certain types of soil and so on. But we wanted to find the gut-derived ones because they have special qualities with colonization and understanding the gut microbiome, right? And so they started sampling lots of different stool and taking actual samples from the mucosa and isolating the spores to figure out which kind of organisms lived in there and what were the healthy versions of spores. And so they isolated these 15, 20 years ago, probably closer to 20 years ago. Once they've isolated the strains from the source, then they go through and they study them genetically to understand what they do, all the capabilities. Do they have toxin genes? Do they have, antibiotic resistance genes, all these things. And then once they characterize and properly isolate the organism, then they start studying them for functionality, which is where we came in, and we started licensing those strains from the university. And we said, okay, we'll do the clinical work on them to see what they do in the body, actually, and how they react with the microbiome and so on. And so that's where we started building the work. Now, where they're derived from now, we have the original cultures, but we do fermentation. So we grow the cultures we use a chickpea flour as a base with amino acids and proteins and all that stuff. And it's a giant fermentation tank, like beer or wine, of course, without yeast. In this case, it's bacteria. So we take a tiny bit from the original seed culture that was isolated, and then we inoculate a small flask. We grow it up. Then we take that flask and inoculate a bigger flask, grow it up, and then you finally put it in the big 10,000-liter tank, and then you grow up a lot of these spores. Wow. And then when you get to a certain density of growth, you stop the growth, you shock them to go into the spore form, because spore bacteria in their spore form are extremely robust, right? They can survive in the capsule for 10 years, a million years, actually. It doesn't matter. The oldest spore that's ever been found was 250 million years old. It was in salt crystals in a cave in Southern California. And researchers have been going into caves, looking at areas that untouched by man, right? Because they're trying to find new antibiotics, right? All antibiotics come from microbes. So they're trying to find new ways of developing antibiotics. They're going into caves, taking samples. They find these crystals, that they dissolve the crystals, and in them are Bacillus endospores, still alive. And they were 250 million years old, sitting there, right? Just in the spore form, just waiting for the right environment to come out of the spore form. And so they can sit there indefinitely. They can survive through the gastric system in the spore form. And then when they get into the small intestine, they do like this little molecular handshake with the lining of your gut. They now know they're back in the gut, which is their natural home. And then that breaks them out of the spore state, and they become like a regular functioning bacteria, right?
Very cool. So is this a product that you would recommend just to the average listener, like as a daily sort of probiotic?
Yes, for a couple of reasons. So number one, when we started the company, again, my big thinking was like, what do we want to solve? What's the biggest problem we want to solve? And what we wanted to solve right off the bat was leaky gut, right? Because intestinal permeability is the biggest driver of chronic low-grade inflammation, which then is the biggest driver of chronic disease. And in fact, a 2015 meta-analysis paper, which means it's a study of a lot of studies on the topic. So it's a conclusive study on the scientific consensus on a topic. They concluded that leakiness in the gut and the translocation of toxins, meaning the movement of toxins from the lining of the gut into circulation, was the number one driver of mortality and morbidity worldwide. Right? It's a number one killer. Leaky gut is the number one killer worldwide, because it's the foundation for the vast majority of chronic illnesses. So when we saw that, we said, okay, we need to solve this as a problem, and microbes are going to be the solution, because it's microbes that maintain the lining of the gut, right? We don't do a whole lot ourselves to maintain the lining of the gut. We outsource that. Another example of outsourcing things to the microbes that live in the environment. And as it turns out, in the world of veterinary medicine, they've been focusing on leaky gut for almost 60 years. And they do it in the context of farmed animals and so on, because they get sick and they die. And a high mortality rate in a chicken farm is a really bad economic thing. And so they've been investing a lot of research to understand why the animals are getting sick, what's compromising their growth, why their immune systems are so weak. Of course, the horrific conditions in which they're being housed is a big driver, but they're trying to figure out what about the horrific condition changes the physiology of the animal, right? And what they're finding in these animals is that all of these conditions create leakiness in the gut. Their exposure to antibiotics, because animals are fed all kinds of antibiotics and vaccines and so on, the poor quality feed that they get, the toxic environment that they're in a hen house or chicken house, for example, there's ammonia everywhere, from the chicken farts and everything. Building of ammonia in a grow house in a chicken is an absolute disaster, because it can actually explode the entire building. That's how much ammonia is built in that environment. Right. So they've been trying to figure out all this toxicity in the environment. What does it do? It actually creates leaky gut. Leaky gut makes the animal sick and weak, and then animal dies. And so they've been using probiotics in the veterinary space to alleviate leaky gut for almost 40 years now. And they can dramatically reduce the mortality rate despite being in those toxic environments. Now, that's not dissimilar from us, right? We use antibiotics a lot, not only directly, but also through our foods, right? We live in toxic environments. We have, gases and carbons and all these glues and all these off-gassing that occurs from all the things around us. We're surrounded by, electromagnetic flux and so on. We eat really poor quality food, and everything around us is actually very similar to those farmed animals. And we have leaky gut as a result, right? And that becomes the biggest driver. So we looked at the analogies there, and we looked at the types of strains. And that's where we came up with another reason why the spores became so important, is because they've been using spores in that environment to alleviate leaky gut for like 50 years. So we said human-derived spores may do the same thing in humans. And sure enough, we published our first paper in 2017 showing that, yes, Mega Spore and the specific spores in Mega Spore absolutely do alleviate leaky gut in as little as 30 days, right? So that became the important part. And that's why spores are so different in that way. They're the only microbes that can do that. Now, I also want to clarify one thing. Not all spores are also created equal, right? You're starting to see other spore-based probiotics coming out. When we first started, there was not a single spore-based probiotic on the marketplace, right? We created this genre of probiotics. Now, because like in the supplement industry, it's a very much a copycat industry. And when companies see something is growing and demand for it is growing, they kind of jump in and try to do an imitation version of it. So now there are companies that are just calling any distributor, buying spore bacteria and throwing it together again, without doing any research. You have no idea if those spores actually colonize. Can they do quorum sensing? What do they do to the immune system? How do they impact the microbiome? They're not all created equal. A 1% difference in the genetic capability of the spore will completely change its functionality, right? I'll give you an example. Remember that bifidobacteria we talked about, the 1714 strain with that EPS coating, right? And how it has all of these fascinating gut-brain impacts. And then we tested it with other bifidobacterium longum strains. And we saw that actually when you mix them, it actually knocks out the activity, right? And these strains that don't have the EPS don't have any of those functionalities. The genetic difference between these two strains is one gene. One gene difference makes this strain super powerful for modulating anxiety, stress, sleep, and all of that, gut-brain neurological inflammation. We didn't even talk about neuroinflammation, but all the neurological inflammatory conditions that this strain helps with, all of those powerful things because of one gene difference to this species that doesn't do any of that, right? So microbes can be profoundly different from one another due to very, very small differences in change in genetics. And so that's another reason why utilization of probiotics or microbes requires a good amount of research to understand what the microbes actually do.
You just unpacked so much there. I have like 5 follow-up questions. I'm not sure if I can remember them, but let me go back to, you were talking about how animals many times are given antibiotics. Humans are also given antibiotics. So is there any sort of case for, back to that, 50 billion strain probiotic you mentioned, do you feel like there is a case for maybe short course of a high burst of probiotics to kind of, I don't know, re-inoculate the gut after heavy antibiotic use, and then to maybe go back to a maintenance of like the Mega Spore?
So there's actually data on this. And in fact, there's 2 big published studies that show that those kind of high concentration probiotics actually negate the recovery of your microbiome. And in fact,
wow, I would not have expected you to say that,
right? Which is completely opposite, right? But what's interesting about it is, one of the things we came out saying in 2013 when we first started talking about the microbiome is that there is an issue with these, high dose, high concentration probiotics that just have lots of different strains in it, competing with your natural flora, right? Because if you take a lactobacillus, acidophilus, for example, and you naturally have acidophilus in your gut, this acidophilus could very well compete for the binding sites with your natural acidophilus, right? Or negate it in some way. And we always talked about that. And the only way to know that it doesn't do that is to do studies on it, and then most of these products have no studies on them.
Mhm.
So we always got the question about what do we do to recover the gut from antibiotics? Do we take a broad spectrum
Mhm.
probiotic? Our answer has always been no. I would hypothesize that it would actually negate the repair. And then sure enough, those two big studies that came out in 2018 and I think 2019 that showed exactly that, that after a course of antibiotics, if you took these kind of high concentration multi-strain probiotics, it slowed down the recovery of your microbiome. In contrast, we did it and we published a study on the spores showing that when you took the spores, not only with the antibiotic, but right after the course antibiotic, it actually improved and accelerated the recovery, right? And here's why. Let me, I think it's important to understand what's happening in the microbiome when you take antibiotics, right? So let's say you have each of my fingers are different microbes in the gut microbiome, right? And they're all at kind of equal level right now. Of course, an antibiotic comes in. What happens when the antibiotic comes in within the first 2.5 hours is all of them get knocked down by over 90%, right? All of the microbes equally. Most microbes in the gut are going to be susceptible to the presence of antibiotics, right? So most of them get knocked down. What then starts to happen is they start to grow back at different rates, right? Now your microbiome, after that first course of antibiotics, starts to look like this. And then the second dose comes in, boom, everything gets knocked down again. But then things come back at a different rate. Now they start to look like this. And multiple doses of the antibiotics, finally, you have all these microbes still, but they're at completely different levels than when they first started, right? One reason for that is the constant changing environment in the microbiome. So an example of that is a healthy microbiome, a healthy gut environment really tends to be more acidic, right? So your stomach, we know, is super acidic, and then the small intestine can be relatively acidic at the proximal side near your stomach, and then typically is around 5.5 to a pH of around 6. And then as you get towards your large bowel, you start to see more neutralization of the acidity in the gut. Now, there are lots of good bacteria in your gut that produce lactic acid. And it's the lactic acid that acidifies the gut and maintains stability. Now, one of the reasons why the acidity is important is because a lot of egregious microbes and fungus and mold, and mostly fungus, can't grow at acidic environments, right? A lot of the pathogens don't grow well in acidic environments. So that acidity in part keeps control of many of the dysfunctional microbes that may overtake the system. So when you take a course of antibiotics, all of these lactic acid producing bacteria get knocked down. That means there's no lactic acid being produced. So now the pH of the gut is a little bit higher. So as the bacteria come back, the ones that do better at a higher pH come back a little faster.
Sure.
Each knockdown, the acidity level, the base level goes up. The gut becomes less and less acidic. And so then the microbes that do better at the less acidic environment come back faster and faster and faster. So what happens when you're done with the course of antibiotics, all the microbes, for the most part, that you had in the beginning are still there. They're now at just completely different levels, right? All these beneficial bacteria that were there before now are at such low levels that they actually are not really functional, but they're there. Right. But these dysfunctional bacteria that are at low levels are now at high levels. And so this is the dysbiosis that occurs after antibiotics. It's not that you're wiping out and eliminating and microbes are gone forever. Right. So that re-inoculation idea doesn't actually work in that sense. What you actually need is something to come in and readjust the levels of these microbes. And the spores do that.
Yeah, kind of to sense what the problem is. And yeah.
Exactly. It's no different than your garden, right? If you haven't tended your garden in a while, the weeds are going to start to grow, the environment's going to change, and the plants that you're hoping to grow are getting suppressed by the weeds and so on. So it's really about going in and making an ecological change in that garden. Same thing in your gut. It's about going in and readjusting the environment so the good guys come back and the bad guys get suppressed, right? And prebiotics and spores, diet, all of those things will help do that.
I initially told you 45 minutes, but we're already at 45. Do you have 15 minutes for?
I do, yeah.
Okay. Just don't want to abuse your time. So is Mega Spore probiotic contraindicated in anyone? Like, who should not take this product?
We haven't found anyone that should not take it. The only people we say are people who are on specific immunosuppressants because of organ transplants, right? If you recently had like a kidney transplant or something like that, and you're on a regimen of immunosuppressants, you shouldn't be taking any supplements anyway, unless you're really working carefully with your doctor,
Sure.
right? So outside of that, there
So
really is.
what about SIBO? What about SIBO? Can we go
Yes.
there? Yes.
Yeah, absolutely. So SIBO people need the spores almost more than anyone else, right? So let's talk about why probiotics have been contraindicated for SIBO people to begin with. Right? So SIBO, really, when you look at from a microbiome perspective, the way you define SIBO is there's a taxa shift. Number one, in the small intestine, you're supposed to have a predominance of gram-positive bacteria. Right, those are the good functional bacteria in the small intestine, and they're not supposed to grow above a level of around 10 to the 4 CFUs per gram of stool in that area, right? So that's a level. The number doesn't really matter, but it's about 10 to the 4. So imagine you have gram-positive bacteria at 10 to the 4. That's a healthy small intestine. Now, for a variety of reasons, and I do a whole SIBO talk for a couple hours, but a lot of it is about stomach acid compromise. So using PPIs, or having overgrowth of H. pylori that compromises stomach acid. You have a lot of gram-negative bacteria in your mouth, Enterococcus and so on. That is the major source of SIBO bacteria, is the mouth, right? The mouth is full of the exact bacteria that seem to dominate in the SIBO gut. And this becomes a daily source of this microbes because you're swallowing billions of those gram-negative enteric bacteria into your gut. But what's happening is under normal healthy conditions, all of these bacteria that you're swallowing are getting killed in the stomach, right? From the stomach acid. So they're not entering the small intestine in a viable state. So if stomach acid is compromised because of H. pylori, or because you're using antacids, so on, drugs, PPIs, right? Then these organisms are getting past the stomach acid in a viable state, and they're colonizing the small bowel, and they're taking over the small bowel. Right. So that's step one of why SIBO is being created. This is why there's something called PPI-induced SIBO. That is a definitive condition. This is why H. pylori infection is also associated with SIBO. Right. Now, the other thing that's associated with SIBO is liver dysfunction. Right. There is, and I'm trying to remember the numbers off the top of my head, but around 50% of people with SIBO also have compromised liver function. When you compare that to same age match individuals who don't have SIBO, it's less than 14%, right? So you have 3 times more likely to have liver dysfunction if you have SIBO, and you're 3 times more likely to have SIBO if you have liver dysfunction. So they go hand in hand. Now, why is that? Well, when your liver becomes compromised, one of the first things that becomes compromised is the production of bile acids, right? The bile acid pool that your liver produces that's stored in your gallbladder and secreted through your small intestine gets compromised. So the bile acid pool shrinks and shrinks and shrinks. So your volume of bile that you can produce and secrete during the consumption process reduces dramatically. Why does that have an impact on SIBO? Well, bile does a very, very important thing to keep the growth of bacteria very low in the small intestine. Number one,
I always tell patients it regulates pathogenic load. Yeah,
it does. Because it's an antimicrobial by itself, right? Bile is an antimicrobial on itself. And through a single meal that you eat, you can actually recycle the same bile acid pool up to 15 times.
Wow.
Right? The bile gets released through the gallbladder into the small intestine. It moves through the small intestine. It gets reabsorbed at the end of the small intestine, goes back to the liver. The liver picks up all the nutrients and toxins and does what it needs to do with that, cleans up the bile, sends it back to the gallbladder for recycling again and release in the small gut, right? So when you have food in that system, you are coating the system with bile. Now, what that does is that means that it doesn't allow the microbes that live there to go, hey, there's food, we can go crazy and start fermenting things and producing gas and creating all of these byproducts, right? Because the bile is there suppressing the metabolic activity of the microbes when food is present. Now, the other thing that bile does is at the very end of the small intestine, when it's getting reabsorbed, it triggers a receptor called the nuclear FXR receptor. That receptor causes the small bowel lining to release antimicrobial compounds. Again, as a secondary protection to make sure that bacteria are not active when food is present, right? And that's a really, really important protection so that you don't get fermentation in your small bowel, which is essentially what's happening in SIBO, right? So when you have bile acid compromise, not only do you have the lack of bile moving through and acting as an antimicrobial itself, you also have bile not activating the nuclear FXR receptor as much, so you don't have the antimicrobial protection that is supposed to be present during food and during feeding. Now you've got these enteric bacteria that are moving in, and they have higher numbers in the gut, and on top of that, when food is present, they have the chance to metabolize and create bloating and gas and hydrogen and methane and everything else that they produce, right? And so SIBO is that dysbiotic. It's a liver function issue. It's an oral health issue because you've got too many enteric pathogens in your mouth. It's a stomach acid issue. And then finally, SIBO causes significant leaky gut. The presence of gram-negative bacteria in the small intestine that's now inflamed because of all the production of ammonia and all these other noxious gases that lining is not used to, right? Now it's leaky and it's inflamed. And because there's gram-negative bacteria, they have something called lipopolysaccharide or an endotoxin. That endotoxin is leaking through the small intestinal lining. And what happens is it goes up the vagus nerve and it lodges itself in an area of the brainstem called the dorsal-vagal complex. When LPS from your gut lining ends up in the dorsal-vagal complex, what happens is the signals from the brain to the gut to move the bowels stop. Now your bowels are in stasis, which is another feature of SIBO. Your bowels don't move, right? The movement of the bowel is so important to control bacterial growth for two reasons. One, because remember, food transits out of the small intestines within something like 3 hours, right? 2.5 to 4 hours-ish, food is basically moved through the small intestine, going to the large intestine, right? That timing is incredibly important and very biologically significant. The reason is because bacteria have something called a lag phase of growth, right? So if you take food, for example, and you set it out on your counter, right? Food that should be refrigerated or it'll overgrow microbes, right? It doesn't overgrow microbes immediately, right? It takes 8 hours, 10 hours, 12 hours to overgrow. The reason why it takes so long, even though that's a good environment for bacteria to grow, is because all bacteria go through a lag phase where their metabolic activity is really, really slow until they all of a sudden hit a logarithmic phase and they go nuts, right? That lag phase is like 7-8 hours. So if the food can transit out of that environment within 2 or 3 hours, the bacteria never go into log phase growth, right? So transit time is really important. Now, the LPS leaking through and creating this bowel stasis means that when you eat food, the food is staying in your small intestine for 8, 10, 12 hours, right? Your transit time is super slow. You're constipated, so you're not even pooping it out. Your things aren't moving through. And then the other part of it is not only is it the peristaltic movement of the bowel that stops, the migrating motor complex signal stops. The migrating motor complex is a sweeping electrical signal that basically cleans out the small bowel, right? And you don't get that. And so you don't have that cleaning effect to make it a food-free environment for most of the day, right? And so those are all the things that compound together to create the condition of SIBO, and it starts with a dysfunction in the oral microbiome, in the stomach, in the liver, and so on. So one of the key things to stopping SIBO is resealing the lining of the small intestine, because if it's leaky, you're going to continue to have stasis. You also need something to go in there and fight some of these gram-negative bacteria, right? Because they're the dysbiotic organisms in the gut. The spores do both of those things. They seal up the lining of the gut. They stop the leakiness of the gut. They can start to trigger bowel movement. They can compete against the gram-negative bacteria and bring their levels down, right? And then they also protect the liver. We've published 2 studies now showing spores have the ability to protect the liver, and people who have SIBO have extreme amounts of toxicity on the liver, because not only do they likely have SIBO because the liver was already being compromised for whatever reason, right? the endotoxins that leak through, 85% of the endotoxins that leaks through goes first to the liver. And then the liver has to deal with all of that endotoxemia, which then leads to non-alcoholic fatty liver disease, right? Non-alcoholic steatohepatitis, and so on. And that progresses along that line, which means that bile acids become more and more compromised. So the liver support is so important. And we've been able to show in published clinical trials that the spores protect the liver. So that's another reason. So
very cool.
Yeah. Very, very long answer to your poignant
You're
question.
Just so smart. I'm
just there's so much there, right?
Totally.
All the dots.
Now you have me thinking, my patients who have had their gallbladder removed, obviously we're supporting, I am supporting their microbiome and giving them oxbile if they need it. But now I'm thinking, oh my gosh, these individuals are at a greater risk of SIBO. I mean, you just kind of made me realize that, which is totally the case. What about patients who, you're saying patients with SIBO should tolerate this, and you made a strong case for why they need these spores for various reasons, but what if they have an adverse reaction? Because I will say I am one of those individuals who's had SIBO, who didn't tolerate probiotics. In the past, I need to try this again, but in the past, I didn't tolerate it. I will say my symptom that I always get is tachycardia. That has just been my symptom. And I'm presuming it's endotoxemia because these are, killing off the gram-negative, like you were mentioning, bacteria. And that's kind of had been my response, which what has really helped me are serum-derived immunoglobulins, which is why I really want to get to that piece also, right? Because you kind of alluded to that earlier, that we also need to bind pathogens and toxins and not let them leak through our gut lining. So I know I'm asking a lot here, but first let me go back to, if someone's not tolerating a spore-based probiotic, why do you think, what's happening?
Yeah.
Like what?
So it's a
is my presumption correct on that?
It is. Yeah. It's a classic Herxheimer reaction. It's a detox reaction. Or naturopaths will call it a healing crisis, right? Where these bad bacteria are being killed off, which means they release their toxins in larger loads, and your body will go through different responses to that. But it's actually the perfect segue for one of our solutions for that. Is it doesn't mean you can't do the spores. It means two things to me. It means you have to go much slower. And then number two, you need something to negate that Herxheimer reaction, which immunoglobulins are the perfect thing for that, right? These bovine serum immunoglobulins are like a magical thing. It's one of the few things that I take like religiously every day, right? Especially when I travel and all that too. It is such a gift to us because think about it this way, and it's largely IgG, but there's some IgM antibodies as well and IgA, but it's largely IgG. IgG are those big, awesome neutralizing antibodies, right? That are very specific to certain things. And imagine these cows are out there in the pasture and encountering all kinds of stuff, right? Environmental toxins, viruses, microbes, pathogens, all that stuff. And their immune systems are building antibodies against all of these things that absolutely neutralize these things, right? Including environmental toxins, which is a very important part of it. And so then we get to pull out the serum from the cows and concentrate the immunoglobulins and utilize them. What we're doing is we're sending in these little amazing drones that have specificity for all kinds of toxigenic things, including mold toxin, viruses, bacteria, right? Environmental toxins, all of these things that they then neutralize all of these things, including LPS.
That you said earlier.
Yeah, they bind that too in the lining of the gut, dramatically bringing down the toxigenic and immunogenic load in the gut. And that allows for a respite in the inflammation and toxicity in the lining of the gut, which then allows the gut to heal, right? And this immunoglobulin by itself has studies on HIV enteropathy, for example. HIV enteropathy is a very severe form of leakiness in the gut in HIV patients. In fact, the NIH published a study showing that HIV enteropathy was a better predictor of mortality in HIV patients than viral load itself, right? Because the leakiness in the gut that they experience drives all kinds of other risk factors, right? Including chronic low-grade inflammation and so on. And then there's also studies on a pediatric ulcerative colitis patients where they're showing the ability to modulate the lining of the gut even under that kind of condition. Then the question becomes, well, how do they actually, repair the damage that's occurring in the lining of the gut? They repair it by allowing repair to occur, right? So the analogy I give people is like, if you get a cut on your hand, it's going to perfectly heal unless every single day you rub it and irritate it, right? Imagine you wake up every morning and you rub and scratch at that cut. It's never going to heal. Inflammation and toxicity that we experience every single day from things coming in or endotoxins is the same as rubbing your cut. So if your gut is leaky and damaged and the lining and the mucosa are all damaged, if there's constant inflammation in the area, it never repairs, right? And that's the same thing that would happen on your hand.
Sure.
So what the immunoglobulins do is they give some respite. They reduce the toxigenic load. They reduce the inflammatory response so that your system can actually repair. That's why they work so well with the spores, because the spores induce repair, but and these and the immunoglobulins negate the inflammation and toxicity, so the repair can actually happen.
And just for the listeners, how they do that is they are binding all of these bad things that you've mentioned, making them so large they can't get reabsorbed into the bloodstream, so you poop them out essentially.
That's exactly
right. So it's lessening that burden on the immune system. But just so the listeners can put that together, it's basically you take these bimouths, you take these immunoglobulins, they bind to those bad things, and then you poop them out. Just had to simplify because you're so smart. I had to just
no, and it makes
break that down. Yeah,
that's a great way to put it. And I do want to distinguish it from other binders, right? Because they're not like
not an environmental binder. Yes,
exactly. Which bind like a bunch of stuff and take it out. These are so specific to specific things, right? A single immunoglobulin that's specific to an aflatoxin in mold will only bind to that aflatoxin in mold, but it's so specific to it that it'll bind to it in such a strong way that it'll never let it go, right? And that's the beauty of these immunoglobulins is they're so specific and so powerful in their capability, but they also don't bind good stuff, which is great, right? Because you don't want them binding nutrients and all your good bacteria and so on.
Totally. I know the serum-derived immunoglobulins have really helped me. So I think that is what's happening. And personally with me, I do want to go back to, you had mentioned again, like vets have done a lot of research. I had no idea that maybe they were ahead of us, but they've done more research on impacting their animals' microbiome, right, for their animals' longevity, whatnot. I want to just bring in really quickly something that I think has helped my animal is that you also have a probiotic for animals. Actually, that's not this is FODMAPE, which I want to get to too, but phytospore. You have phytospore. Yeah. Can you briefly just mention kind of what organisms are in that? I had a holistic vet who I interviewed on this podcast once tell me that it actually had, I'm going to probably murder this, but something like a similar makeup to some organisms that are found in grass, because sometimes animals are like my dog will go and eat grass sometimes. And so she's obviously, her body's trying to get something. Yeah. So is that correct? Or can you expand on that and the strength of that sort of product that you carry for animals?
Yeah. It was our belief that our animals, so when you look at our animals, and my dog just passed away a couple of weeks ago, my 13-year-old, right? Everyone was heartbroken about it. And the poor thing had like 2 different types of cancers and, and then when you think about that and you go, how often does cancer and hip dysplasia and diabetes and all that occur in the natural animal kingdom, right?
It doesn't
at all, right? And there's some evidence that it doesn't. They've looked at this thing, and yet our dogs, our pets, our cats suffer from our same kind of diseases, right? And my hypothesis was that it's probably because they live in our environment, which causes leaky gut, right? And so I started looking for information on leaky gut in canines or in felines, and I didn't really find much. And so we set out to prove that our pets suffered from leaky gut and that the leaky gut for them was equally impactful to their overall health as it was for us, right? They had endotoxemia. And so that's what we discovered, that sure enough, they have endotoxemia, they have LPS. It does create significant inflammation that leads to lots of other conditions. And so for us, we're like, okay, if spores can help us, maybe they can help them as well, but we probably need something else in addition to that, right? So we picked 2 of our spores that are known to also exist in animals. So Bacillus subtilis and Bacillus coagulans. And then the third organism that we picked is something called Pediococcus acinolactici. Pediococcus is the organism that's found in grasses. Okay. Right? And there's actually 2 very natural environments for Pediococcus. It's found in grasses or shrubs and things like that. You find it in environments. And humans have also been using them for fermenting sausages for thousands of years, right? And that also was developed from exposing sausages to the environment. right? This organism ends up on the sausage because you're carrying it in your sacks or whatever, and then they ferment the sausage in such a way that it creates a new flavor. And so we've been using for the last couple thousand years Pediococcus even till this day in commercial sausage production as a way of fermenting sausages for improved flavor with Pediococcus, right? And that's probably the reason why dogs go and they lick the ground and they eat grass when they're not feeling well. They're trying to get this organism in. This organism is a powerful immune modulator in dogs and cats, right? It dramatically reduces the inflammatory response and the allergic response in dogs. That's why it helps a lot with atopic dermatitis in
Well, my
dogs.
dog was licking her paws. That's why, yeah. I mean, I changed her
that's
food. The first thing
absolutely-
I did was change her food. Yeah, she's actually eating raw dog food. I don't know how you feel about that, but I've changed her
No,
to
it's
raw
great.
and also put her on the phytospore and some also supplements to mitigate histamine and whatnot. But yes, that was literally one of the reasons I sought out that probiotic, which we didn't previously have on the shelf and we do now here at the Integrative Health and Hormone Clinic.
Yeah, and that's
So,
that's the number one health condition among dogs and to some degree cats as well, but dogs for sure is atopic dermatitis. You know, they have allergies and atopic dermatitis. And that's all because their microbiomes are ruined. And so the Pediococcus plays an important role, and there's some good studies on that. So then when we mix the combination of Pediococcus and Bacillus, because that's the things they would encounter in the outside environment, we were able to prove and publish a study showing that not only did dogs have leaky gut, but then we can also resolve the leaky gut in as little as 7 to 8 days by using this combination of bacteria. And that's where the phytospore came from.
very cool. Okay, I will wrap this up. I could talk to you for hours, but I will start wrapping this up here. I do want to go back to I already shared a little bit of my story, how I have history of SIBO, whatnot. I will say, and for the most part, my symptoms have greatly improved. I've done a lot of gut work, but I will say one product that changed my life, just the amount of foods that I can consume, is FODMate. I can literally go eat Brussels sprouts, and they're just foods that I had to be so cautious with before, kind of just knowing my limit. So can you also talk about, this is not a probiotic, so we're off the spore topic now. This is different, but this is an enzyme. Can you kind of expand on what this is for, the ingredients as well? And it's literally changed my life.
Yeah, it's a phenomenal product. And to me, I was quite surprised when it didn't exist already, right? Because you know, when we got into this world of gut health and I started looking at, you know, the practices that we do and the therapeutic gaps that exist in people's conditions, one of the things that really made me nervous was this idea of a low FODMAP diet, right? And a lot of people have to do it, unfortunately, because the resulting symptoms of consuming FODMAPs are really you know, debilitating in many cases, right? It goes all the way from just an annoyance of having bloating and discomfort all the way to being debilitating for some people. So understandably, people then just avoid them. The problem with avoiding them is FODMAPs tend to be food categories that lots of your good beneficial bacteria like in your large bowel, right? But because of your dysbiosis and dysfunction, you're getting them fermented in your small bowel, at least some components of the FODMAP. So what we said is, wow, there's enzymes that exist that actually break down those components that would normally be fermented in the small bowel and allow them to move down to the large bowel without being fermented in the small bowel. That gives you both the opportunity to tolerate the food, but then still utilize the benefits of the root components of the food.
Get the nutrients. Yeah.
You know, and so to us, it's a healthy microbiome is all about bringing back resilience and foods into your system, right? If you feel okay by eating just 7 things, that's not a good condition to be in, right? You need to have some degree of resilience and be able to figure out a way to introduce things back into the system. And that's where the FODMate enzyme came from. So we looked at some of the key enzymes that are required to break down the carbohydrate structures in FODMAP foods so that they don't produce gas in the small bowel. And we put those enzymes in, and fortunately, there were already studies on these enzymes. And the way we did it, we partnered with a naturopathic doctor on this who does a lot of work with SIBO. We formulated the product. She took the first 50 bottles or so and tried it with a bunch of patients. And they all came back, and just like you were saying, they were like, wow, this is a game changer for me. I can eat things I couldn't touch for years now. And that is so much joy to us. And we're like, okay, we got to get this out. This is
Oh, it's like amazing. Every time it goes on back order, my husband, who's our office manager, he's like, "Don't worry, I set a bottle aside for you because I can't run out of this stuff." So,
That's awesome.
for the listeners who have had SIBO or who have had fructose intolerance, right? Those who haven't been able to tolerate the high gas producing foods, the FODMAPs he was alluding to, this enzyme, when taken with meals, is just it is amazing. It's awesome. So thank you for that. Yeah.
Welcome.
Yeah. Let's see, what else should I ask? I think we've covered most everything that I wanted to today. You're just a wealth of knowledge. I'm sure you've heard that a million times, but is there anything else you want to share that we didn't go over? I mean, we covered so much. Is there anything we're missing that you feel like you want to share?
Yeah, I do just want to mention, we mentioned H. pylori, right?
Sure.
And I do want to emphasize that H. pylori, to me, is probably now one of the biggest dysbiotic drivers in our gut microbiomes. And it starts at a very early age. Most people, over 50% of adults have infectious levels of H. pylori, and most of us got exposed at an early age, 7 or 8 years old. And since that time, H. pylori has been driving dysbiosis in the gut. And in fact, now it's tied to lots of different conditions like autoimmunity, infertility, reflux disease, of course, right? Gastritis, gastroparesis. That's the lack of emptying of the stomach, right? That feeling really hard fullness in the stomach once you eat. And then the bloating and the distension in the stomach after you eat. So that inflammation, all of these very common digestive symptoms are all driven by this pathogen, including very serious things like gastroesophageal cancers and gastric cancers. And then, even things that are seemingly unrelated, like autoimmune disease and infertility. And so for us, H. pylori is a big issue that has to be tackled. And in the allopathic world, they have something called triple therapy, now quadruple therapy, which is 2 antibiotics or 3 broad spectrum antibiotics and a PPI, right? Which is probably the worst atom bomb for your gut and your microbiome you can think of.
Yeah, that's a good analogy. I've never heard of that as the atomic bomb for your gut, but that's good.
Atomic bomb, right? We're reducing stomach acid and we're destroying the microbiome, the microbes all at once with 3 broad spectrum antibiotics and a proton pump inhibitor. And so for us, we're like, there's a better way to do this, right? And so we found a microbe. This is a natural microbe. We haven't done anything to the microbe. And this is where the ghost probiotic or the postbiotic comes in. Because this microbe, it's a Lactobacillus reuteri that has a significant affinity on its cell membrane structure for Helicobacter pylori. It actually, all it does, and this is the only thing we've been able to see that it does, is it binds H. pylori, it co-aggregates with it, and it strips it out of the body and takes it out through defecation. And it's not even alive, right? This is where, so you'll hear terms like ghost probiotic or postbiotic, which describe microbes that don't have to be alive to elicit certain functions, right? So this microbe is intact, but not alive. You take it, you basically open the capsule, you mix it in water. And the reason you do that is we want to coat the stomach nicely with the bacteria. So you drink it, it coats the stomach, it grabs Helicobacter pylori and takes it out. We have 10 published studies on this particular organism and its ability to reduce Helicobacter pylori. That, I think, for you as well, and most patients, you'll start to feel a difference almost immediately, because you don't realize how impactful Helicobacter pylori is in the symptoms you experience every single day with the bloating, the distension, the lack of bowel movement and all that. You'll start to feel it from the very first day.
Did you say that's Lactobacillus rhamnosus or reuteri, or what did you say?
It's
reuteri. I always said reuteri. I've always associated that with women's health. That strain.
That's the beauty of probiotics, right? 1 or 2 genes different, and it has a completely different functionality.
So this is like how am I saying that? A same strain, similar strain, but different gene. Is that what has slightly different?
So it's same species, but different subspecies, right?
Got it. Okay.
So a microbe. So you can have a Lactobacillus, which is a genus, and then reuteri, which is a species. And then you can have subspecies of reuteri that all function differently, but they're all still reuteri because they share about 80% of the same conserved genome, right? So there are certain sections of a microbial genome, they're called these hypervariable regions, that if they have all of those regions and they're in the same group, right? But within the same group, they can have completely different functions,
functions,
right? like E. coli, for
Yeah.
example, right? You can have
Yeah.
E. coli 015787, which if you get exposed to will make you sick as a dog and maybe hospitalized, or you have up to 100 different E. colis that naturally live in your gut that never do anything bad, right? So the
Yeah.
difference is only 1 or 2 genes. So this is a reuteri that is specialized at binding Helicobacter pylori. That's all it does.
Yep.
And then it takes it out of the body. It's
And what's that product called? We don't
so
have that
that's
product.
called PyloGuard.
That makes sense.
PyloGuard.
Okay.
Yeah, I actually have it. I have a bottle here because I just ordered a bunch. This is one that I take regularly as well, because so this is what it's called.
But if you took that and the Mega Spore, those aren't negating each other's function, like you're saying some other probiotic. Okay.
Not at all. Yeah, this one you take once a day, or in some severe cases twice a day, and you just mix it in water. I take it on an empty stomach. That's the best way to take it for me. It's one of the first things I do in the morning. I take that with a couple of the supplements I take on my empty stomach.
Wonderful. Thank you for sharing that. I'm sure listeners are going to want to hear more from you. So where can listeners find you?
Yeah, well, absolutely. They, reach out to me on Instagram. My handle is Kiran Biome, so KIRANBIOME. I post a lot of my interviews and talks and all that on there, so you can access them that way. If you look on YouTube and you put my name, just Kiran Krishnan, you'll find tons of interviews and all that have been uploaded kindly by other people. And then certainly come to our website, Microbiome Labs. We have resources there as well. And there's a new site that we're building that's actually built out. That's a incredible resource for microbiome information, and that's called Better Biome. Better Biome is a site that we've created that's just, and it's got different microbiome clinical experts. They put articles and videos and all that on many different topics on the microbiome. There's no products on there or anything like that. It's all about just resources and information. So you can go on Better Biome and search for resources as well.
Awesome. Thank you. We'll post links to those in the show notes. And then last question, finally, we're here. What is your top longevity tip?
Yeah. So my top longevity tip is building resilience, right? I don't want to be healthy by having to make 100% the right decisions. I want an 80-20 rule. I want to make 20% bad decisions that are fun and still be perfectly fine, right? That to me is quality of life. And the way you build resilience is by building a very resilient, healthy gut, because your gut is what allows you to have exposures to things that aren't really great for you, but still deal with them. Your gut, facilitates the vast majority of detox pathways, right? Your gut facilitates repair of your cells. We talked about urolithins earlier, right? So if something damages your mitochondria, your gut will produce urolithins to fix it right away. Your gut induces autophagy, or which is a way of cleaning up broken DNA and damaged cells and all that, so you can recover better. Your gut protects your liver, your gut protects your pancreas, your gut protects your gallbladder. So it protects your brain. So it is the central command center for the functionality of your body. And if you have a resilient gut, you have a resilient system, and that allows you to live a really thriving, healthy life without having to make 100% the right decisions.
I love saying mic drop. Yes, that was so good. So good. Okay. Great way to conclude the show. So thank you so much for coming on today and just sharing what wonderful things your company's doing, and for your dedication to the study of our microorganisms in the gut. You are brilliant. Again, I could listen to you for hours. This was excellent. So thank you so much.
It was my pleasure. Thank you so much for having me.